So now I'm going to talk to you about vancomycin resistance. So in bacteria, from a medical perspective, there's two broad classes of bacteria. So I'm going to write here, vancomycin resistance. So you can write this in your notes. Vancomycin resistance. Okay. So there's two types of bacteria. From a medical perspective. So we're talking medically now. So from the perspective at your doctor's office and the perspective of a medicinal chemist, there's two types, and those are gram-negative and gram-positive. You've probably heard those terms before. So gram-negative and gram-positive. So the difference between gram-negative and gram-positive in the most simplest case is that gram-negative uptake, but they don't retain gram stain. So they uptake, but they don't retain gram stain. I'll show you what gram stain is in a second. So basically, gram stain goes right through them. Gram-positive bacteria uptake and retain gram stain. So what is gram stain? So gram stain looks like this. And it is purple. And so what you do to test whether a bacteria is gram-negative or gram-positive is you do the following test. Here's the test for it. You take your bacteria and you plate it on a petri dish. So you grow your bacteria out on your petri dish. You take a sample from somebody's throat or something for strep throat or whatever. You plate your bacteria. So you're going to plate your bacteria out on a petri dish. And so you're going to stain with crystal violet. That's the same thing as gram stain. You're going to stain with this gram stain or crystal violet. And so what's going to happen is your cells are going to turn purple. So all your cells are going to turn purple because they're all kind of, now they've uptaken it. And what we're going to do next is we're going to wash them. So the ones the gram stain can kind of uptake, but they don't retain it, that it can go right through them. Once we wash it, they're going to get rid of their gram stain. Those are gram-negative bacteria. The gram-positive bacteria are going to hold on to the gram stain and stay purple. So we're going to add in some ethanol, and that's going to wash away from the cells that can be washed their gram stain. So this is going to wash. So we have an ethanol wash. And so what we'll get there, if you've like all of us, have a lot of different bacteria in your body, on the surface of your skin, you're going to get some bacteria are purple. So some bacteria are purple now. But all we can really see, if we were to look at this on a microscope, were that some bacteria are purple. We wouldn't be able to see the other bacteria that couldn't hold on to the gram stain. So we need to do something with those. So what we use is what's known as a counter stain, and so that's going to make the bacteria that right now are colorless show up. And so you can use something like fuchsin or safranin O. So I'm going to draw safranin O here for you. This is all really commonly done. This is pink. So we're going to counter stain. So we're going to counter stain with this dye safranin O. We're going to counter stain these cells. And so the ones that are able to hold on to that ground stain are going to stay purple, and the ones that couldn't hold on to the gram stain are now going to turn pink from the safranin O. So we're going to have a mix of purple and pink bacteria. So all the purple bacteria are gram positive, and the pink are gram negative. And so why do we care? What does this tell us medicinally? Why is this important? Well, what it tells us is that for gram-positive bacteria, it tells us something about the outside of the bacteria. And based on what it tells us about is the nature of the cell walls of the bacteria. And based on that, we can give the bacteria different drugs. Gram-negative bacteria respond to different antibiotics than gram-positive bacteria. So for gram-positive bacteria, what you're going to have basically is you're going to have a thick cell wall made of what's known as peptidoglycan. So it's basically peptides and sugars together. So we're going to have a peptidoglycan cell wall that's thick. So this is what's on the outside. It's this thick peptidoglycan cell wall. Then what's going to happen is we're going to have a space. So we're going to have this as a periplasmic space. We're going to have a space here. And then we're going to have the plasma membrane. And then inside here is where all the bacteria's enzymes and DNA and all that kind of good stuff is. Remember, because bacteria don't have an enveloped nucleus and stuff. So, but this is where all the good stuff's happening, where all the biochemistry is happening, is just inside here. And this thick cell wall retains gram stain. Now for gram negative bacteria, what we're going to have is an outer membrane. The outermost layer is a membrane. And this outermost layer is a combination of a lipopolysaccharide and protein. So we're going to have, for gram negative, we're going to have an outer membrane that's a lipopolysaccharide and protein. So it's like a lipid-based membrane, and there's proteins and sugars involved in that. Then we're going to have a periplasmic space. And then we're going to have a peptidoglycan layer that's thin. So we're going to have our periplasmic space. And then I'm drawing in there another peptidoglycan layer. And then there's going to be another periplasmic space. That's going to be another periplasmic space there. And then you're going to have the plasma membrane. And then inside that plasma membrane is where all the good stuff, all the bacterial enzymes, DNA, all that stuff is. And so what happens was when you wash the gram-negative cells with ethanol, it breaks up that outer membrane, and that peptidoglycan layer is too thin to retain the gram stain, and so the gram stain comes back out. So the ethanol wash step breaks the outer membrane, and that thin peptidoglycan cell wall won't retain gram stain. And so for cells that are gram-positive that have this thick cell wall on the outside, what we can do is we can break it up. And the reason that we can break it up and deal with these gram-positive cells is what their cell wall looks like. So their cell wall, and you can actually do this with gram-negative cells too, to a certain degree depending on the drug that you use. So cell wall in bacteria, what it looks like, but it's especially exciting. The bacteria cell is gram-positive. So it's super deadly then to inhibit cell wall biosynthesis. So what the cell wall looks like a lot of times, so I'm going to draw, basically, it's going to have a number of sugars. So I'm drawing in, I'm starting to draw in a polysaccharide. So what I'm drawing in it on the top here is a polysaccharide. So that's glucanac, and this is N-acetyl glucose, and this um, right here is um, an N-acetyl muramic acid. And here we have attached to that sugar, we have an alanine, and then we have a D-glutamine, and that through a lysine is going to be attached to two other D amino acids. So remember, all our amino acids are L, but here's a D amino acid, and then through a lysine. So this is L-lysine. I'm abbreviating a polypeptide here. And that's going to be attached to that lysine. There's going to be two D-alanines. So we have two D-alanines linked together, and this is always the case in these cell walls. As part of the biosynthesis, they have these two D-alanines linked together. And so we don't have D amino acids. And so this is a D amino acid linkage to top it all off. And so this is a great place to go after with a drug. And so that's what they do with vancomycin. Vancomycin is going to mess up this D-ALA linkage here. It messes up the bacteria's ability. It messes up anything in your body's ability to make this D-ALA linkage. And so that means that the bacteria can't make their cell wall. And so that can be really devastating. And then it doesn't hurt you because you don't have any D-amino acid cell walls in your body, nor other than your natural microbiota that live in your body. But in your human cells, you don't have this.